Okay, thank you, Doctor.
All right. Record. Okay. So this is we know that when we discuss when we look at this on the subject, metabolism of lipid and membrane metabolism, lipid and membrane metabolism. We know that we have two bigger classes. We have lipids, on the metabolism itself and also on membrane. Okay, so now because membrane and lipids is we know that membrane, the structure of membrane itself is lipid. Okay, so you can see from this slide, on the difference between triacylglycerols, and also membrane lipids. Okay, we know that lipids, triacylglycerol fatty acids, so basically meant for energy storage. Storage lipid. For energy. But for membrane lipids, so it can either known as phospholipid or also glycolipids, which phospholipid it will form glycerophospholipids and sphingolipids. But same goes to glycolipids. We can synthesize sphingolipids from glycolipids. So let's look at the difference in terms of component, in terms of the structure between triacylglycerols and also on the membrane lipids. As you know, the structure of triacylglycerols, we have 3 molecules of fatty acid that are esterified to glycerol as a backbone, for triacylglycerol. The structure of triacylglycerol. So what is the difference, of glycerophospholipids, compared to triacylglycerol? For glycerophospholipids still have, glycerol as the backbone, and it has 2 molecules of fatty acid, and by looking at the name of the molecule glycerophospholipid shows that it has phosphate group and also alcohol group at carbon 3. All right, but for sphingolipid, instead of glycerol as a backbone, it has this sphingosine molecule that act as a backbone for sphingolipid. All right, but still it has 1 molecule of fatty acid, and also phosphate group, and also one example, it has choline, that attach, to form this sphingolipid. There are, later we are going to discuss, there are 4 major groups that can attach to this phosphate group of sphingosine. We have choline, we have ethanolamine, we have inositol, and also we have serine. So there are 4 major groups that can attach to sphingosine to form sphingolipid. Sphingolipid that falls under glycolipids, still it has the sphingosine as a backbone. It has sphingosine as a backbone, and it has 1 molecule of fatty acid, but by looking at the name of glycolipid, it tells us that it has either mono or oligosaccharide that attach to carbon 3 of sphingosine. To the sphingosine backbone of glycolipid. So it has this mono or oligosaccharide. So that's differentiate between sphingolipid that falls under phospholipid and also sphingolipid that falls under glycolipids. All right, so triacylglycerol. So now let's go back to triacylglycerol, on the biosynthesis process. Biosynthesis process. So it it can occur in adipose tissue. We know that in adipose tissue is the the location where it has been, kita punya fat being stored, okay, dekat adipose tissue, okay, for energy, okay, for energy, okay. And also, triacylglycerols can be synthesized in the liver, which it is mainly secreted as VLDL, and it can be transported. Ingat tak, during your first test, saya ada tanya kan, okay, how VLDL, mana tu? Soalan ni saya tak ingat dah. Okay, all right. Triacylglycerol is synthesized by esterification of fatty acyl-CoA with either glycerol 3-phosphate or dihydroxyacetone phosphate. Okay, you still remember this molecule, DHAP? Okay, one of the intermediate dalam
Glycolysis.
Yeah, okay, dalam glycolysis. So basically, this, okay, the synthesis, the biosynthesis process of triacylglycerol, basically it generates phosphatidic acid. So this phosphatidic acid, or the other name of phosphatidic acid, which is diacylglycerol 3-phosphate, okay, is one of the key intermediate in the biosynthesis of triacylglycerols and also other glycerolipid. Okay, this is the key intermediate. Okay. All right. Okay, so the major building block for the synthesis of triacylglycerols, okay, in tissues other than adipose tissue is glycerol 3-phosphate. Okay, so basically adipocytes. So the glycerol 3-phosphate is the precursor, okay, for the biosynthesis of triacylglycerols in other tissues, but not in adipose tissue. Why? Because adipocytes, adipose tissue, like this enzyme, which is glycerol kinase. Okay, so that in adipose tissue, it uses this dihydroxyacetone phosphate, which is produced during glycolysis as a precursor for the triacylglycerosis in adipose tissue. Okay, so you need to know why is it adipose tissue cannot utilize glycerol 3-phosphate as the precursor for the biosynthesis process. Okay, so it tells us that adipocyte must have glucose. Okay, because we know that dihydroxyacetone phosphate is the intermediate in glycolysis. So you need to have glucose to oxidize in order to store fatty acids in the form of triacylglycerols. Okay, now let's look at this pathway. Okay, so we know that we have these two. Okay, we know that in adipose tissue, okay, in adipose tissue, it requires dihydroxyacetone phosphate, okay, which it can undergo reaction, okay, that catalyzed by glycerol 3-phosphate dehydrogenase, okay, to produce glycerol 3-phosphate. So basically, in adipose tissue, it, okay, is still need it still need glycerol 3-phosphate, but okay, it need to be produced from DHAP. Okay, okay, but in other tissues, okay, it can produce glycerol 3-phosphate from glycerol, which undergo phosphorylation that catalyzed by glycerol kinase to form this glycerol 3-phosphate. Okay, all right. Okay, so glycerol 3-phosphate, if you look at this, okay, at these reactions, it undergo two acylation step, okay, which involve the transfer of acyl group, okay, to this glycerol 3-phosphate. Okay, if you look at this diagram, so basically you the fatty acid is activated, okay, is activated to fatty acyl-CoA. Okay, so this acyl group is transferred, okay, to carbon 1, to carbon 2 of glycerol 3-phosphate, okay, to form this phosphatidate. Okay, phosphatidate. Okay, so this is the fatty acid being, okay, it undergo activation, okay, catalyzed by acyl-CoA synthetase to form fatty acyl-CoA. So this acyl-CoA, this acyl group is transferred by acyl transferase. So the first acylation step, okay, it transfer the acyl group to carbon 1, okay, and the second acylation step, okay, so it transfer this enzyme transfer the acyl group to carbon number 2, okay. So the first this is the formation, the steps involved in the formation of phosphatidate or phosphatidic acid. Okay, so basically this is the summary. Okay, you can have a look. Okay, so basically phosphatidate. Okay, if you look at the structure, okay, phosphatidic acid, or also known as 1,2-diacyl-glycerol 3-phosphate. Okay, that's the specific name. Okay, 1,2-diacylglycerol 3-phosphate. Okay, by looking at the structure of a phosphatidate. Okay, in most phosphatidate, okay, the fatty acyl chain attached to carbon 1 is saturated. Okay, while the one attached to carbon 2 is unsaturated. So basically, we can try to find, okay, examples kan. Okay, apa contoh phosphatidates that have carbon 1 saturated fatty acid, okay, and carbon 2 unsaturated fatty acid. Okay, but okay, so this is the theory behind this. Okay, in most phosphatidate, carbon 1 saturated, carbon 2 is unsaturated fatty acid chains. Okay. Okay, I forgot to mention, okay, the first acylation step produce lysophosphatidic acid. Okay, the second acylation step produce this phosphatidic acid or 1,2-diacyl-glycerol phosphate or 3-phosphate. Okay, in order to now we want to synthesize triacylglycerol. Okay, so this phosphatidic acid, okay, undergo hydrolysis, okay, and the reaction catalyzed by phosphatase enzyme, okay, phosphatase enzyme to produce diacylglycerol. So basically this step remove, okay, phosphate group, okay, from phosphatidic acid. Now we are left with diacylglycerol. Okay, now kita nak synthesize triacylglycerol. So you need to undergo another acylation step catalyzed by acyl transferase. Now we get triacylglycerol. Okay. All right. So the key intermediate, okay, yang paling penting ni lah, okay, phosphatidic acid. So you need to know, okay, the structure, okay, of phosphatidic acid, how it can be synthesized from either from glycerol 3-phosphate or from dihydroxyacetate phosphate. And from phosphatidic acid, how can we get triacylglycerol? Okay. Okay, so this is another diagram to show how can we produce triacylglycerol from phosphatidic acid. Okay. All right, so phosphatase, okay, enzyme that remove phosphate group. Okay, now we left with 1,2-diacylglycerol. Okay, then okay, is another acylation step which it involve addition another acyl group to form this, the addition of acyl group at carbon 3 to form triacylglycerol. Okay, so later, okay, we will discuss on the so this is basically the head group, okay, the head group that can attach to other molecule. Okay, contoh serine. So this is basically polar head group that can attach to other molecule. Contoh serine, choline, ethanolamine. Okay, serine, choline, ethanolamine, we have inositol. Okay. All right, now, okay, we have, we have discussed on the biosynthesis of triacylglycerol. So one of the intermediate is phosphatidic acid. Okay, phosphatidic acid. So phosphatidic acid, from phosphatidic acid, we can synthesize triacylglycerol. Okay, it's well known on the characteristics of triacylglycerol on the non-polar part, hydrophobic, neutral lipid. Okay, and from phosphatidic acid, we can synthesize glycerolipids. Okay, we have phospholipid, we have glycolipids. Okay, so this is the key intermediate for the production of both triacylglycerols and membrane phospholipid. Okay, so glycerolipid basically is a membrane lipid. Okay. All right. So that's okay. We are done with glycerol biosynthesis of triacylglycerol. Now let's move to glycerolipid biosynthesis. Okay, glycerolipid biosynthesis. Okay, now we are going to discuss more on membrane lipids, on phospholipids. Okay, so okay, the most abundant phospholipids are made from glycerol. Okay, we know that. Okay, we know that glycerol has been the precursor, okay, for the formation of phosphatidic acid. So it is very important, okay, in the glycerolipid biosynthesis. So phospholipids are important components of membranes, okay, and also it helps in transport triacylglycerol and cholesterol in the body. So basically, it's mainly for membrane. Okay, even though membrane lipids, okay, there are so many roles, okay, of membrane lipids in our body, either in brain, either in organ protection, either in blood clotting. Okay, there are so many important roles, okay, of membrane lipids. Okay, so glycerolipid biosynthesis, the biosynthesis process occurs mainly primarily in endoplasmic reticulum. Okay, so okay, phosphatidic acid is made either into the acylglycerol. Okay, we are going to discuss on this later. Okay, so okay, kita dah discuss ni, glycerolipids are synthesized by phosphorylation and acylation of glycerol. Okay, now this is the structure of glycerolipids. So basically, okay, you punya 1,2-diacylglycerol tadi, phosphatidic acid, dia lebih kurang sama. Okay, you punya phosphatidic acid punya structure. Okay, you punya acyl group, R1 mainly saturated, R2 mainly unsaturated. Okay, it has almost similar structure to triacylglycerol. Basically, this is a glycerol-based lipids. Okay, let's say, okay, so this is the head group. Okay, so there are other molecules that can attach, okay, to this head group. Okay, if let's say hydrogen that attach, okay, that replace this X, okay, it will form 1,2-diacylglycerol. Okay, if phosphate group that attached, okay, here, so this structure known as glycerophospholipid. Okay, if carbohydrate that attach, okay, to this structure, to this head group, it will form glyceroglycolipid. Okay, carbohydrate, glycol, phosphate group, phosphol. So that's how the naming of the molecule, basically it explains on the structure. What do you have in the structure? Okay, so either 1,2-diacylglycerol, we have glycerophospholipid, we have glyceroglycolipid. You can have a look, okay, on the component that attach, okay, to the molecule that attach to this head group. Okay. Okay, let's look on what is glycerophospholipid. Okay, so this is the most abundant, okay, for the most abundant in life organism. This is the main lipid component of biological cell membrane. Okay. Okay, it has other function. You can have you can do your own reading on glycerophospholipid. Okay, I just briefly put in some of the important role of glycerophospholipid. Okay, so the other function, it helps in dissolving triacylglycerols and cholesterol. Okay, in a lipoprotein form. All right, so the major class of naturally occurring phospholipid with phosphate containing head group. Okay, so basically glycerophospholipid basically is a polar lipid. Dia dah lain dia bukan, it doesn't break the characteristic of lipid. Okay, because of that phosphor group, now it becomes polar lipids. Okay, so it can produce from 1,2-diacylglycerol from phosphatidic acid. Okay, when phosphate group attached, okay, at the head group, it will form this glycerophospholipid. Okay, so what makes this glycerophospholipid as a polar lipid? Because okay, it has this polar component because of the phosphate group. Okay, and this phosphoryl group can link to another group. Okay, it can link to another group. Okay, okay, it can link to another group. And kita dah ada now. We have this glycerophospholipid now. Okay, from phosphatidic acid, we have this glycerophospholipid. But this head group can link to another group, which is these are the 4 major polar components that can bind to phosphoryl group at carbon 3. Okay, either water, okay, either ethanolamine, choline, serine, and also inositol. Okay, okay, how do you differentiate the structure of this, of ethanolamine, choline, serine, and inositol? Which one is amino acid? Dalam banyak-banyak this polar component kan, which one is amino acid?
Ethanolamine.
Okay, ethanolamine. Okay, so now you already know. So basically, if you look at the structure of ethanolamine, choline, serine, okay, inositol ni saya tak letak structure. So basically, inositol, the structure is quite different because it has a ring structure. So lagi senang nak ingat. Okay, so ethanolamine basically, only ethanolamine is amino acid. Okay, in this when we compare between these 4 polar components, and if you look okay at the structure of ethanolamine, choline, and serine, basically all these 3 polar components have this nitrogenous base, kan? Okay, they have this nitrogenous bases. Okay, kalau serine, we have this carboxyl group. Okay, so that's how you differentiate between these polar components. Okay, so let's say if a water molecule that attached, okay, to this structure, okay, it will form this phosphatidic acid. Okay, 1,2-diacylglycerol phosphate. Okay, if ethanolamine, okay, that link to the phosphoryl group at carbon number 3, so now we have this phosphatidyl ethanolamine. Okay, this phosphatidyl ethanolamine. Okay, so what is the importance of phosphatidyl ethanolamine? Okay, so it can be found particularly in nerve tissue and also spinal cord. Okay, as a polar head group, it creates a more viscous lipid membrane. So the viscosity of the lipid membrane is very important. Okay, the viscosity of the lipid membrane. Okay. So basically, glycerophospholipid, we know that it is a polar lipid, so it's highly amphiphilic. Okay, highly amphiphilic. All right. Okay, so this is phosphatidyl ethanolamine. Okay, I have read somewhere in humans. Okay, so this phosphatidyl ethanolamine is important. Important untuk kita punya heart. Okay, untuk kita punya heart. Okay, but the mechanism, I can't remember on the mechanism. Because when the blood flow to the heart is restricted, so this phosphatidylethylamine has its important function, okay, to protect the heart, to mungkin facilitate. I don't know, I can't remember the mechanism, but I read somewhere that this phosphatidylethylamine is very important when we have problem, okay, with the blood flow to the heart, okay. And also it play an important role, okay, selain pada dekat nerve tissue and spinal cord, so it plays an important role in blood clotting as well, okay, dekat blood clotting, okay, because it increase the rate, okay, it increase the rate, it works together with phosphatidylserine to increase the rate of thrombin formation. Kita tahu kan dalam blood clotting we need thrombin, okay, to help in the to stop, okay, to clot the blood. So it works together with phosphatidylserine, so it increase the rate of thrombin formation. So it has their importance in human, okay. All right, so if choline that attached to the carbon triphosphorate group of glycerophospholipid, now we have this phosphatidylcholine. Okay, so it's a major component of cell membrane. So basically semuanya untuk cell membrane. So it can be easily obtained from diet. Okay, we can get it from egg, we can get it from soybean. Okay, cari lagi what are the sources of choline, phosphatidylcholine. Okay, and also it has it plays a role in membrane mediated cell signaling and also enzyme activation. Okay, it's being called as a miracle molecules. Okay, I want you to do your own reading on this miracle molecules. Okay. Okay, so this is the phosphatidylinositol tadi yang I mentioned just now. Okay, inositol, it has this ring structure. Okay, when inositol attached to the phosphate group, so we have this phosphatidylinositol. Okay, so for membrane basically, so it also participates in many metabolic processes in either in plants and also animals. Okay, so basically from glycerophospholipid we can generate, okay, other derivatives molecule of glycerophospholipid, which is phosphatidylethylamine, choline, serine, inositol. Okay, so these are the derivatives of glycerophospholipid. Okay. Okay, now let's look on the synthesis of this derivative of phosphatidylcholine and also phosphatidylethanolamine. So we know that this PC and PE are the most abundant phospholipid in eukaryotic cells. It's very important for membrane. Okay, so for the biosynthesis process, okay, the pathway basically they use choline and ethanolamine obtained either from the diet of the body's phospholipid. Okay, so in the liver, phosphatidylcholine also can be synthesized from phosphatidylserine and also phosphatidylethanolamine. So let's look on the synthesis process of phosphatidylcholine. Okay, so basically you need to know what are the basic units for phosphatidylcholine. Okay, dia ada glycerol, dia ada fatty acids. Okay, so this is the biosynthesis, the pathway, the reactions involved in the biosynthesis of phosphatidylcholine. Okay, the first step is the phosphorylation of hydroxyl group of choline or ethanolamine. Okay, now let's discuss on both. of choline dengan ethanol amine, formation of phosphatidylcholine or phosphatidylethanolamine. Okay, so it's involve phosphorylation of this hydroxyl group. Kalau phosphorylation require ATP. Okay, so now it produces this phosphocholine. Okay, all right. Okay, if it is phosphorylation of ethanolamine, so now it produces phosphoethanolamine. So these are the enzymes that catalyze this reaction. Okay, ethanolamine kinase or choline kinase. Okay. All right. Then, okay, we have, okay, now we have this phosphoethanolamine, kan, and also phosphocholine. All right, so this structure, okay, this phosphoryl group, okay, this phosphoryl group now attacks this CTP. Okay, CTP stands for cytidine triphosphate. Okay, instead of ATP, now it attacks CTP, which is cytidine triphosphate, which it displacing, it remove one phosphate molecule, okay, to form this CDP ethanolamine or CDP choline. Okay, so enzyme that catalyze this reaction is phosphoryl ethanolamine or phosphorylcholine cytidyl transferase. So basically, it involves the transfer of cytidyl, okay, cytidyl cytidyl group, okay, to this phosphoethanolamine. So take note that the CTP is activated intermediate. That's why phosphoryl group attacks the CTP and form CTP ethanolamine or acetylcholine. Okay. So you need to understand on the mechanism what happened. Okay, just not mention. Okay, it's being converted to the but without explaining. Okay, what happened. Okay, on the mechanism. Okay, part. Okay, then final stats. Okay, so CDP ethanolamine or CDP choline now is going to react with 1,2-diacylglycerol, which this hydroxyl group attack this phosphoryl group of CDP ethanolamine and now forming phosphatidylethanolamine, and it remove this cytidine monophosphate. Okay. So the hydroxyl group attacks the phosphoryl group of CDP ethanolamine. So it remove CMP and now it formed. Now we get phosphatidylethanolamine and also phosphatidylcholine. Okay, so there are 3 steps involved in the biosynthesis of phosphatidylethanolamine or phosphatidylcholine. Okay, you all okay?
Yes, doctor.
All right, okay. So this is a simple diagram to show on the reactions on the phosphatidylcholine biosynthesis pathway. Okay, all right. So I love this diagram. Okay, so now nak ingat it highlighted the important, okay, component that involve, okay, that will be transferred or that will be removed, okay, in this pathway. Okay, now let's look on how phosphatidylserine can be synthesized. So basically, it can synthesize from phosphatidylethanolamine, which involve the exchange of the ethanolamine group dengan free serine. Okay, simple je. Okay, all right, to produce phosphatidylserine. All right, so but phosphatidylserine, when it undergo decarboxylation, it will give, okay, it will produce phosphatidylethanol amine. Okay, so this is how the exchange of ethanol amine group dengan serine group, okay, in the phosphatidylserine synthesis. Okay. Okay, a little bit on synthesis of phosphatidylinositol. Okay, okay, CDP diacylglycerol produced from phosphatidyl acid combines with inositol to form phosphatidylinositol. Kat situ macam is a direct reaction, okay, to form phosphatidylinositol. Okay. Okay, take note that this PI, it contains arachidonic acid. Ingat tak arachidonic acid ni berapa carbon? Hmm?
20.
Okay, okay, so 20 carbon unsaturated fatty acid. So it, okay, it has, okay, this PI, it has this arachidonic acid on carbon 2. Kita tahu kan carbon 2 biasanya dia ada unsaturated fatty acid, okay, which serve as a substrate for prostaglandin synthesis. Okay, arachidonic acid also act as a precursor for cholesterol biosynthesis. Okay, next kelas nanti kita kena discuss on cholesterol biosynthesis. Okay, so kita akan jumpa balik this arachidonic acid. Okay, so this PI is important for signal transmission across membrane. Nanti dengan kelas Dr. Izwan, we are going to discuss on all this mechanism dalam signal transmission, how this lipid membrane plays an important role dalam nerve, signal transmission, neurotransmitter. Okay, there are many more. Okay, so that one on Dr. Izwan punya part. Okay, remember the structure of phosphatidylinositol. Okay. All right. Now we have discussed on the synthesis of triacylglycerol, on synthesis of glycerophospholipid, on the phosphatidylethanol amine choline kan, serine inositol. Now let's look on the phosphatidylglycerol. Ada lagi satu. Okay, so basically dia lebih kurang sama, is nearly the same, but it involves only in two reactions. Okay, attack of the carbon 1 hydroxyl group of glycerol 3-phosphate on the CDP diacylglycerol yielding phosphatidylglycerol phosphate. Okay, so to get this phosphatidylglycerol, so you need to remove the one of the phosphoric group through hydrolysis, okay, to form this phosphatidylglycerol. Okay, and the condensation of 2 molecules of phosphatidylglycerol, okay, we will get this cardiolipin. So that cardiolipin. Okay, what is cardiolipin? Okay, all right. Okay, so how can, okay, this one, okay, from CDP diacylglycerol, okay, we can synthesize phosphatidylinositol, ni lagi clear, okay, and we also can synthesize phosphatidylglycerol. Okay, so basically, kalau if you compare, if you notice, okay, the difference between this pathway dengan pathway untuk synthesize phosphatidylcholine ethanol amine tadi. Alright. If you notice that kalau dekat sini ni sebenarnya hydrophobic part yang activated. Dia attack dekat hydrophobic part. Alright. Rather than the polar head group. Okay, contoh dia akan attack dekat hydroxyl group at carbon 1. Alright, okay. Glycerol triphosphate, okay, they attack to the hydroxyl group of carbon 1 to form this phosphatidylglycerol triphosphate, then it undergo hydrolysis. Okay. Okay, so this is the structure of glycerol phospholipid, okay, in one page, so that you can see the difference. In terms of structure, okay, kalau inositol dia ada, it has this ring structure, phosphatidylcholine or lecithin. Yang mana satu yang structure apa yang nama dia? Yang ada, maksudnya. Okay. Kalau you tengok diphosphatidylglycerol or cardiolipin, it has this structure difference. Okay, dia ada dua main structure. Okay, on left hand side, left and right. Okay. Ada soalan? No, tak ada. Okay. Can we continue?
Yes,
Yes.
doctor.
Okay. All right. So from triacylglycerol, we have moved. Okay, kita dah discuss on glycerolipid, glycerophospholipid. Lagi satu tadi? On the synthesis of phosphatidylethanolamine, choline, serine, inositol, and also phosphatidylglycerol. Okay. So now let's move to sphingolipid. Kita tahu, okay, we know that sphingolipids, if we compare to triacylglycerol, it have sphingosine as a backbone compared to triacylglycerol. Okay, dia tak ada glycerol as a backbone. Okay. So basically, what makes sphingolipids interesting molecule? It has this complex amino molecule, which is sphingosine. Okay, it's more complex lipids. Okay, they are a very long chain. Okay, carbon 18 and more than that. Okay. So basically, it's a derivative of sphingosine. Sphingolipid is a derivative of sphingosine. So it's important for nerve cell, membrane cell. Okay, so basically, okay, sphingosine. Okay, when there is fatty acid that attached to it, to the amino group dekat sini, it is known as sphingolipid. Okay, sphingo, okay, from sphingosine, okay, the backbone of sphingolipid. Okay, sphingolipid, it is also known as an acylsphingosine or ceramide. Okay, are you familiar with ceramide? What do you know about ceramide? What comes into your mind when you, okay, bila you jumpa ceramide?
Moisturizer.
Okay,
Lotion.
moisture, yeah. Okay, what else? Okay, ceramide. Okay, but the specific name, so now you know what is ceramide. Okay, so now this is the structure of ceramide. Okay, so the The basic name is N-acylsphingosine. Okay, N-acyl. So it has this. Okay. So ceramide, okay, is a family of lipid molecules. It's important for cell membrane. It acts intermediate in the metabolism of sphingolipid. Okay. Okay, so this is similar to the glycerophospholipid. Okay, so it has this head group. Okay, other molecules can attach, can link to it. Okay, and when, let's say, carbohydrate that attach to this ceramide, so it will be known as sphingoglycolipid. Okay, sphingoglycolipid. Okay, nampak tak dia ada structure, dia ada nitrogen. It has this amino group, nitrogen amino group dalam structure sphingosine. Okay, sphingolipid. If phosphate has the phosphate group that link to the ceramide, so it will be known as sphingophospholipid. Okay, sphingophospholipid. Okay. Okay, so this is the biosynthesis of sphingomyelin. So I'm sure, okay, you are familiar with sphingomyelin. Biasa kan, myelin. Sphingomyelin. Okay, so this is a major lipid membranes of nerve tissue. Okay, this is very, very important. Okay, a very important component dalam kita punya nerve tissue. So now let's look on how this sphingomyelin synthesized. So this is the pathway to synthesize sphingomyelin. Okay, so sphingomyelin, so it's a sphingosine-based phospholipid. Okay, okay, sphingosine-based phospholipid. Okay. So let's look at the pathway. So basically it started with palmitoyl-CoA, which it undergo condensation. Okay, palmitoyl-CoA berapa karbon? Ingat lagi tak? Palmitoyl-CoA berapa karbon fatty acid?
16?
Ah?
16.
16. Okay, so palmitoyl-CoA, okay, it undergo condensation with serine. Okay, undergo condensation with serine, which in this reaction, okay, coenzyme A and also carboxyl group, this carboxyl group and also coenzyme A is removed. Okay, they have been removed, okay, along the way in the reaction, which it is NADPH dependent reaction, okay, to produce this sphingenin. Okay, in order, okay, so sphingenin, so basically the precursor are palmitoyl-CoA and also serine. Okay, so you can see kat mana dapatnya that amino group tu kan. Okay. Okay. So from sphingenin, okay, sphingenin, it undergo acylation, okay, with fatty acyl CoA, okay, acylation with fatty acyl CoA, and it acylate dekat amino group. Okay, acylation, okay, it transfer the acyl group, okay, to this amino group of sphingenin, okay, to form ceramide. Okay, so it involved the conversion of FAD and F2 FADH2. Okay, so this is how ceramide can be synthesized from palmitoyl-CoA. From sphingenin, it undergo acylation. Okay, acylation dekat mana? Dekat amino group. All right, so it formed now ceramide. Okay, so this is the acyl group. Okay, from ceramide, okay, to synthesis, okay, it undergo another reaction, okay, to form sphingomyelin. Okay, ceramide, okay, react with phosphatidylcholine, okay, and we get sphingomyelin. Okay, sphingomyelin. Okay. Okay, certain books, it has this dia lagi details tau. Macam this one is simplified. What what I mean by simplified meaning that okay, in the formation of sphingolin ada certain books that it divided into two. Okay. Maknanya condensation of palmitoyl CoA and serine, it will produce keto 3-keto sphingolin. Okay, then another step, okay, undergo this NADPH dependent reaction baru produce sphingolin. Okay, this one dia dah simplified jadi satu. Alright, but basically it involves different two different enzymes basically kat sini. One synthase enzyme, another one reductase enzyme. Okay, synthase, condensation, okay, reductase enzymes also because it's an NADPH dependent reaction kan, so it requires this reductase enzyme, okay, to produce sphingolin. Okay, all right, then so basically same goes to this step. Okay, in the formation of ceramide, certain books they divide into two. Okay, from sphingolin. Okay, mungkin first step it involve the formation of dihydroceramide. Okay, before it can maknanya first one is in the first step involve the transfer of acyl group, and followed by the second step baru involve FADH2 dependent reaction before it can form ceramide. Okay, but this is simplified version. You can use either way. Let's say kalau if you referring to other textbooks, okay, it is not wrong. Okay, if you want to elaborate more on the reaction, okay, in the synthesis of sphingomyelin. Okay, all right, so we know that from sphingosine, okay, we can synthesize other derivative of ceramide. Contoh, okay, examples, let's say ceramide, okay, with a carbohydrate polar group, okay, we can synthesize either cerebrosides and gangliosides. Okay, familiar tak dengan cerebrosides and gangliosides? Kat mana biasa you jumpa?
Otak.
Kan? Otak. Biasanya dekat TV dalam iklan apa?
Susu.
Susu for newborn babies, for kids, kan? Cerebrosides and also gangliosides. Okay. Okay, so what is cerebrosides? Okay, so this is the simplest form of sphingoglycolipids. Okay, simplest form of sphingoglycolipids. All right, so ceramides, okay, ceramides, when it has this sugar residues attached to it, okay, which is monosaccharides, okay, it will form this cerebroside. Okay, contoh glucocerebroside. When there's a sugar, it will form this glucocerebroside. Okay, so this is the biosynthesis of cerebrosides lah. Okay, either gluco or galacto. Okay, Kalau sugar to glucose is glucose cerebroside. If it is galactose, is galactocerebroside. It depends, okay, on what type of sugar that attached to the ceramide. Okay, how about ganglioside? Okay, ganglioside. Okay, when oligosaccharides, okay, that link to the ceramide, so now we form gangliosides. So this is one of the most complex lipid. Okay, so it is related kat kita punya brain nerve. Okay, we know that we have so, okay, can you imagine, okay, what do we have in our brain? How this, okay, how they, let's say lah, kita punya apa, how does it, okay, how fast, okay, the information being passed from brain to kita punya organs. Okay, contoh, kita punya spontaneous reactions, okay, we have touched something hot, kan? Okay, brain can pass, okay, the information that us, so kita punya, okay, kita punya tangan akan terus tarik daripada benda panas. So how fast and how efficient and how complex the system in our brain that helps us to like, we can do our daily course, we can do our daily routine without any problem. So you can imagine how complex the system in the brain. Okay, I'm really have a very high respect to the doctors, especially in medical lines, okay, that have to do all these operations. They're trying to understand. I'm sure every cases that came to them will have is a different cases, so they have to really sit down, understand, and decide on what they need to do. Okay, because they are working with now, so they really have to. I really have, okay, they really have my highest respect. Okay, if when we learn something like this, okay, we understand more. So it's not that easy, okay, for them, okay, especially doctors, okay, in order to decide, in order to do anything operations, to decide what are the best methods, what are the best procedure, okay, working with brain, working with heart, okay, our body system is very complex. So be grateful what, okay, you are in a good health, okay. So I think we have to be grateful every day. Okay, there are many unfortunate people out there. Okay, we can go to the universities, we can go, okay, we can do whatever what we want to do. I know you are now in a World Cup fever. Okay, I'm sure you have so much fever at the moment. Okay, World Cup fever, you will have final exam fever, kind. All right, so but prioritize, okay, the important part lah dulu, okay. So now we know that okay, what are ceramides, what are cerebrosides, what are gangliosides. Okay, still what is sphingomyelin? Okay, sphingomyelin is a very important membrane that surrounds, okay, nerve cells, axon. Okay, I'm sure you know what is axon. Okay, the importance of nerve cell. Okay, Spine, okay, apart from nerve, I think spine is another important structure that you have to really jaga. Okay, everything. Okay. Okay, because that's give us because it support most of our organs. Okay, most of our organs. So if you that's why you need to have a very good posture. Okay, I think habit. Okay, daily habit will contribute to the bend of your spine. Okay, for example, so that train to have a very good posture, train to have enough rest. Basically, it is important, okay, for our body. Lain lah, okay, lain lah for those who have problem with spine, with any organs from baby. It's called congenital. Do you know what is congenital? Okay, any problems that have been discovered from your from newborn. Okay, maknanya there is certain organs is not developed well, certain or is being detected during first few weeks. Okay, so it's called congenital. Okay, congenital problem, either congenital heart problem, congenital scoliosis, congenital banyak congenital. Okay, so that one is out of kita punya control. Okay, so yeah, all right. So sphingomyelin is one of the important lipid, okay, that very important in play in role in nerve cell. Okay. Okay, let's have a look, okay, on the similarities between phosphatidylcholine and also sphingomyelin. Okay, both have this choline structure. Okay. Okay, one of the important sphingolipid, okay, it can define human blood groups. Okay, so basically human blood groups. Okay, kita ada berapa blood groups, class? Do you know your blood group?
Yes, doctor.
Okay. All right. How many blood groups do we have? A, B, lagi?
AB.
A, B, and O. Yeah. So which one is the universal donor?
O.
Oh, so which one is the universal recipient? Recipient.
AB.
All right. So, I hope, okay, each one of you know what is your blood groups. Okay, so basically, what is the procedure? Okay, the procedure that to determine the blood groups, okay, is based on the carbohydrate moieties, okay, dekat surface of the blood. Okay, because there is a carbohydrate moieties, okay, that helps to determine the blood group. Okay, if I'm not mistaken, kalau galactose, if they can detect, okay, galactose, so it tells us that you have they are the B antigen, so maybe B blood groups B. Okay, I can remember. So maybe you want to read more on how this the importance of sphingolipids, okay, can determine the human blood groups. Okay, kalau untuk A macam mana? Okay, maybe if you have this N-acyl something, okay, that determine A blood group. Kalau B, galactose. Okay, Yang lain, okay, saya dah tak ingat dah. Alright, maybe you want to read more on that, okay? Okay, so there must be a specific carbohydrate, sugar on the surface of the red blood cells. All right. Okay, so now let's move to membrane glycerophospholipid. Okay, membrane glycerophospholipid. Okay, you still okay? Do you want to take a break? 1:10.
Break, doctor.
Okay, break for 5 minutes. Okay.